So this is the first part of a two-part audio lecture for chapter 20. In this part we'll be talking about cell communities and tissues. In the second part we'll be talking about stem cells and cancer. So for our tissues we'll be talking about the extracellular matrix that cells will produce and connective tissue that holds these cells, tissues together. We'll talk about the epithelial sheets, some of the orientation of them, and then the cell junctions that keep them together. So we want to keep in mind that multicellular organisms are a collection of organized tissues or organized collection of cells, right? So tissues are just collections of cells that are organized so that they can provide their function. So for epithelial cells, what we see are that they're cells that are closely bound in a sheet. And so we can see on the image on the right how we have this layer of epithelial cells. And one of the interesting things with these epithelial cells is that we can see the nuclei, which are in red, and they're all around the same region of the cell. Okay, so with epithelial tissues, there will also be an extracellular material that gives it its underlying layer that allow for it to have its structure to shape. Okay, so connections between those epithelial cells and the extracellular material, the extracellular matrix, allows for it to be orientated in the fashion that it is. Okay, so the extracellular material will be the basal lamina, and we'll look at another figure looking at this. Okay, the connective tissue that underlies the epithelial tissue are going to be they tend to there's not as much cells. There's more of the connective tissue, like collagen, that is going to make that up. So we can see in the dark purple color where our connective tissue is, and again this kind of holds the epithelial tissues together. So this shows you the typical tissue. So this is in the gut. So looking at this figure and working from the top down, at the top, this is where we would have our lumen of the gut, and then we see that layer of epithelial cells with the microvilli, the villi that are formed in the gut. So you can appreciate how if we were to take the left side and the right side, we could pull that and elongate that, and we would have this straight line of epithelial cells. But traditionally in the gut, to allow for more surface area and for more absorption to occur, we see these villi that are formed, these finger-like projections. Okay, so underneath that epithelial layer, we can see that connective tissue. So that connective tissue within there, we do have some fibroblasts present. So the fibroblasts are going to be important in orientating the connective tissue so that it does provide that support to it. And then we have additional fibers, so these circular fibers that are present with our smooth muscle. So you'll actually notice that we have two layers of smooth muscle, and they're running in different directions. So we have ones that are running kind of parallel and then longitudinal. So this allows, if we think about, again, the gut and the function, we have to move material through. Smooth muscle we don't think about, so we don't think about like, ooh, gotta move my lunch through my intestines. So the smooth muscles are involuntary movement, and by having circular ones, they act almost like sphincters. When they contract, they cause a pinching in, a narrowing of that lumen. So material is actually propelled forward. Longitudinal allow for movement downstream of the gut. This is where if I was in front of you, could give you a better visual of how this works. But by having those two layers, it allows for that movement of the material more effectively. So if we didn't have circular the smooth muscle orientated for circular fibers, we wouldn't have the same movements, just as if we didn't have the longitudinal fibers. Now, underlying that, we see another layer of connective tissue, because we want to make sure that these smooth muscle cells have that support, that material to anchor themselves. And then we have an additional epithelial layer on the outside. So this would be what would be in the abdominal cavity. So, if we think about a surgeon cutting into the abdominal cavity, they would actually see that epithelial layer that is on the bottom of this graph. They would actually have to, make an incision into the intestines to see the epithelial cells that are line the lumen of the gut. But you can see the complexity of, even a simple tissue like our gut. Okay. And again, some of the things that you should take home from this figure is how the connective tissue, there are cells present in there, but they're not, it's more of a matrix material that makes up the connective tissue. So the cells are there to support the connective tissue, but that's their function. Their function isn't absorption like we see with the epithelial layer. So this just gives you a better view of that extracellular matrix that's found in the connective tissue. So again, we can see these fibroblast cells that are present. We're also going to have immune cells like macrophages. We're also going to have capillaries, so blood vessels that are feeding this connective tissue and the cells that are present in it. But it has a lot of fibers, okay? And so we have these collagen fibers, elastic fibers that are present. We also have these glycoproteins and protoglycans that are present, and all of these fibers and these proteins and carbohydrates are going to end up making like a gel that again is going to provide support as an underlying underlying support system for our epithelial layer, and then on the other side our smooth muscle. So we're going to look more at the extracellular matrix. So as I mentioned, collagen fibers are present into this matrix. The panel A is looking at collagen fibrils in tadpole skin. So we can see how this has a very it almost looks like plywood. So if you did a cross-section of plywood, it's wood fibers that are compressed together. And so these collagen fibers are actually compressed together on the tadpole skin. In figure B, we can see, if we look at the bottom of that figure, you can see the collagen fibers running from the left to the right of that figure. In the middle, we have this fibroblast that's present, so we can see the nucleus there, indicating that that would be a cell. So again, that cell's going to help align those fibers so that they're most effective at providing that support system. But we can see how the collagen fibers are made up of. They're made up of these single polypeptide chains of collagen, and then those come together to form a triple chain, and then a fibril, and then those fibrils come together. So very much like a rope structure. So if you remember back when we were talking about proteins at the beginning of the semester, these alpha helixes coming together, and you see these coiled structures like with keratin, so similar to that idea, and they end up coming together to make a rope-like structure that allows it to be much stronger than if it was just a single fiber on its own. So as I mentioned, the cells that are found in the connective tissue are going to help to align the extracellular matrix, those collagen fibers, so that they can provide the most support, so that they have to be able to deal with the tension, the shifting, the movement of the tissues. So I've mentioned this before when we were talking about the gut. The gut has to be flexible, so it has to be able to deal with increased material, like not a swelling in like a edema type sense, but material moving through, so it needs to be able to flex and expand, but then also contract back down. And so again, having these collagen fibers, this connective tissue underlying being part of the gut tissue helps for that to happen. So these extracellular matrix, these fibers that are present are going to allow cells to be able to attach to them. So you're going to see how those epithelial cells can attach this extracellular matrix. Okay, and so there's fibronectin proteins that allow for that connection. So we can see here on our figure that we have fibronectin. So if you look at panel C, fibronectin has cell attachment sites where they're going to attach to integrins that are present on our epithelial cells. So we can see how we have this integrin present that's attached to the plasma membrane, and then we have our fibronectin having these attachment sites, but they also have collagen binding sites. So they're going to bind to the collagen fibrils that are present in the extracellular matrix that would be at the bottom of our epithelial cell, right? So this is kind of like taking it and flipping it, right? So if we were to think back to the gut for panel C, it's kind of flipping it. So at the bottom of panel C is where we would have our epithelial cells. It would have the actin filaments, adapter proteins, allowing it to bind to this integrin. The integrin has a transmembrane portion, but then also has an extracellular portion. That extracellular portion would bind to the fibronectin. Fibronectin allows for the attachment, so it's kind of like plectin. Inside the cell, we see the plectin makes attachments between the intermediate filaments and the actin filaments. And so, again, the fibronectin is going to have that same kind of role where it's making these connections between our epithelial cell and our extracellular matrix. So we just want to keep in mind that integrins can switch from an inactive to an active conformation. So in the inactive conformation, it wouldn't bind to fibronectin. So it potentially, if you had all these integrins on the basal side layer of the epithelial cells, if those became inactive, it would allow for that cell to dissociate, to release from the tissue, or you could get separation. So you can imagine if someone had a mutation or was exposed to a drug that deactivated the integrins, that you would have a sloughing off of that epithelial layer in the intestines. Okay. In its active form, it's going to be able to bind to the cytoskeleton, as we see on the right side of this figure, and it also binds to that extracellular matrix, to the fibronectin that's going to bind to the collagen. So the extracellular matrix is also made up of protoglycans and GAGs or GAGs, and so these are actually going to form aggregates together. And so here we can see how we have our aggregates forming present. And so these actually act as like a space filler. So we're going to have the fibroblast present, we're going to have our collagen present, but there's going to be a lot of additional space taking up. And so on the right panel here with our electron micrograph, it looks like they're really large, right? But that's because it's kind of zoomed in, right? So they are smaller, but again, they would end up taking up any of the space in which we don't have those fibers present, and we don't have the cells, again, the fibroblasts being the support cells, but also like immune cells and blood vessels and such. So in looking at the gut, we were looking at columnar epithelial sheets. And so there are other arrangements, as I alluded to, and so this just shows you some of the different arrangements that are present. And so for this figure, the yellow you're seeing is where our extracellular matrix would be, our basal lamina. And so we end up having this kind of orientation where we have this apical and basal side of these epithelial sheets. Okay, simple epithelial sheets will have a single layer to them, where stratified will have multiple layers of epithelial between the lumen or the space on the apical side and then what's on the basal lamina on the basal side. Okay, so again, simple one layer, stratified is going to be multiple layers. And then when we look at a simple one, we can further define it by whether it is columnar. So columnar are going to be very tall. They do tend to have villi, microvilli present, right? So villi would be a larger structure that's made up of the epithelial sheet going up and down, like we saw in the gut. Microvilli are those like little hair-like projections created by actin, and you can kind of see those on that drawing of those columnar. And those would be traditionally what we see in the gut. We can also have cuboidal. Cuboidal are going to have a more of a cube or square shape to them. And then squamous are squashed. So they're going to be flat, kind of like pancakes. And so if you've taken A&P and you've talked about the different organ systems, you would have talked about the different types of cells that are present in different organs, in different areas of the organs, depending upon the function. And so I always love the kidney because the kidney is one of those locations where you can actually find all these different types of cells, of epithelial sheets, and it all relates to where you are in the kidney and what the function of that region is. So in an area where we need to have filtration, like the glomerulus, we're going to see the squamous because that actually allows for better, there's actually fenestrations inside the cell, so that it allows for material to go into the Bowman's capsule and get caught. Where then if we're looking at areas on the duct, they're going to be cuboidal because we need some material to be able to go across, but we want to be more selective of that. And if all of this went whoosh, that's fine. It's not on the exam. Just be aware that there are different types, and you should be able to identify the difference between columnar, cuboidal, squamous, and stratified. Okay, and maybe it's helpful to know an example, but that's not necessary because A&P is not a requirement for the course. So with epithelial sheets, they're intrinsically have a polarity, and what that means is that they have an apical side versus the basal side. So the apical side is going to be on the free space. A lot of times this is like the lumen, if we're talking about the intestines, and then the basal side is where the basal lamina is going to be with the connective tissue on the other side. So again, by saying intrinsically polarity or being polarized, it's that it has a top versus a bottom, if you will. And if we're talking about the gut on the apical side, this is where material is going to move through. We're going to have absorption and material entering the cell and then exiting the cell on the basal side. And so if you remember back when we were talking about transport of glucose, and we talked about how we had our SIM transporter on the apical side, and then we had our GLUT transporter on the basal side. Again, that would be an application and where we can see how we have this polarity where we have certain transporters, the SIM transporter on the apical side, and then we have a uni transporter on the basal side. So this just shows you a nicer drawing of the intestine. So we want to keep in mind with the intestine, it's not just epithelial cells that are going to be present there. We're also going to have cells that have other functions besides the epithelial cells that will be absorbing. So we have goblet cells that actually produce mucus. So they secrete out mucus that actually gives a protective layer across the lumen, and so that mucus protects it from any kind of acids and enzymes that are present in the lumen of the gut. It also allows for antibodies to be more effectively trapped in it, so that your gut is part of your immune system, because you'll have antibodies, and if the antibodies were just free floating in there, you wouldn't actually get good contact time with material moving through and those antibodies to potentially warn the immune system that there's something potential that it needs to respond to. Okay. There are other types of cells that are in the intestines, like tough cells. I think I've mentioned tough cells. So tough cells are responsible for taste and smell, but tough cells are also found in your gut, and they act as chemoreceptors to sense different material in there. But they also act to activate the immune response, so that if certain material is found in the gut and taste by the tough cells, it will actually activate the immune system to help assist in responding. So they're really important for parasitic infections. So not just bacteria, but also parasites and parasitic worms. Okay. So again, don't think of the gut as just these absorptive cells that are there to absorb glucose. There are other cells present in the intestines. So in order to keep these epithelial cells connected, there are a number of structures that are present that allow for anchoring and communication between these cells, so that it's not a single epithelial cell present, it's a collection of cells in the tissue, and there is a bit of they respond together. If one senses something, the others can also respond to that, even though maybe their receptor on their cell did not actually sense it. Okay, so these cell connections fall into 4 different broad classes. There are anchoring junctions, adhesion junctions, channel-forming junctions, and cell signaling junctions. So we'll talk about each of these in the slides coming up. So first we'll look at tight junctions. So tight junctions make it so that if something wants to go through to that connective tissue, to the blood vessels that are on the other side of that epithelial sheet, it needs to go through the cell. Okay, so the tight junctions actually form this leak-proof connection between epithelial cells. Okay, so we can see here in panel B, what they've done is they've actually put in this tracer molecule, this dye, and first in the first panel in B1, what they've done is they put this dye on the lumen side. Okay, so we can see these microvilli structures, these finger projections, which are actin, present, and the dye is staying in that lumen area. It's not able to get through. It doesn't leak in between the epithelial cells. So if the molecule was actually to enter the cell, it would have to be absorbed by the cell. Okay. In the other panel, for panel B, so B2, what they've done is they've released the tracker, the dye, on the basal side. And again, you can see how the tight junction stops it from going to the apical side. So tight junctions are almost like a zipper that forms along that apical edge or in between these cells, so that, again, it adds a connection between these cells. Okay, and it excludes things from being able to move along or leak through in between the cells. In addition to the tight junctions, we also have these adhesion junctions, and so these are formed from linking the cytoskeletons through these cadherins. And so, as you can see in the figure, just below our tight junctions, we have these structures that are formed by the cadherins, and we have these bundles of actin filaments that are attaching to them. The cadherins act as Velcro between the cells. So we'll look at that structure next. So here we can see the cadherins, and you have we have these two cells that are coming together, and so we have cadherins that are shown in green and cadherins that are shown in blue from the two different cells, so the different colors of from the different cells. And so again, these kind of Velcro together and hold together. So it's kind of like, you know, if your fingers linked together, and they're coming from different cells. So this gives additional strength to the cell because we have these tight junctions, and then we have these adherents, junctions by the cadherins. So cadherins also play an important role when we're looking at development. So there's cadherin dependent signaling that occurs that actually causes tissues to invaginate, or so as you can see here, this kind of like budding in, turning in on itself, so that they actually will form epithelial tubes. So if you take developmental biology, you will talk more about adhesion, cadherins, and their role in allowing for the formation of the gut and the neurotube formation. So catenin is what allows for the cadherins to have attachment to the actin cytoskeleton. So if we kind of zoom in on this adhesion belt, these adhesion junctions, what we would see is we have those actin filaments coming in that are inside the cell. So we're intracellular inside the epithelial cell. There's going to be anchoring proteins that allow catenins like beta or Ch20 to make the connection with the actin filaments. And then the catenins are then going to connect to the cadherins, which have a transmembrane and intracellular portion, but also have that extracellular portion that will make a connection. Okay, so interesting to note that beta-catenin also functions in the Wnt signaling pathway, so it has a dual function. So, you know, if someone had picked this for the pet protein, you would already know that. But just sometimes proteins have dual functions, and so this would be one that does. So cadherin proteins can also connect to intermediate filaments, and this is what happens in the desmosome. So the desmosome is similar to these adhesion junctions, except that we have intermediate filaments attaching to it. Another structure that we see are gap junctions. So gap junctions are made up of connexins. So these connexins are arranged to actually create a pore. So multiple connexins come together to make a connexon that when open, it has an actual channel in it. So that channel can allow for ions to move through. So calcium fluxing can happen between cells. And so it allows for these tissues to actually communicate with each other. So again, this intercytosolic communication. And again, usually this is because ions are moving through, and that can perpetuate like a calcium flux and allow for responses by neighboring cells. Okay, so in my undergrad, I actually looked at gap junctions in the reproductive system and how this actually allows for this synchronizing of the cilia beating and moving the oocyte along the epithelial, along the oviduct or the fallopian tubes. So I looked in cat tissue, so it would be oviducts. In humans, we call them fallopian tubes. So this figure is a nice figure because it summarizes up these cell connections that we would see in these epithelial sheets. So again, we have these inclusion junctions or tight junctions, and so they're going to form a seal or a waterproof barrier that doesn't allow for material to leak from basal to apical side. It has to go through the cell. At the same time, things can't go apical to basal without going through the cell. We'd have adhesion or adherent junctions. These are going to have actin filaments attaching through cadherins, with catenins playing a role too. Desmosomes, this is where we're going to have intermediate filaments attaching. So again, keep in mind the role of actin filaments and the role of intermediate filaments are a little bit different. So desmosomes tend to have a more of a, allows the flexing of the cell, because intermediate filaments allow that flexibility, that shearing force that could happen to the cell. Those allow, so our adhesion junctions and desmosomes are cell-to-cell connections. So they have two sides to them. I'm going to jump down to the cell matrix anchoring junctions. So we also have hemidesmosomes, where we only have one, and we have these acting linked cell matrix adhesion anchors. It's a mouthful just to say that we have these half, so they don't have the full cell to cell where we have two sides to it. They only have one, meaning so hemi, and that's because they're connecting, embedding into the Velcro, if you will, attaches to the basal lamina to the connective tissue. Okay. So then we also have the channel-forming junctions, these gap junctions, that allow for passage of ions from cell to cell, and allows for the cells to actually respond in a more synchronized fashion. So that concludes this first part of Chapter 20 audio lecture. As I mentioned, there's a second audio lecture that you should listen to on stem cells and cancer. So taking some of this information and applying how cells communicate with one another in the tissue, or miscommunicate when we're looking at cancer. So thank you very much.